So let us discuss our today's topic. In the last lecture, we discussed about the nomenclature of enzymes. Now you know enzymes are named according to International Code of Enzyme Nomenclature, formulated by International Union of Biochemistry and Molecular Biology. So I hope you remember that according to ICEN, the name of enzyme should have two parts. One part should indicate the substrate, another part should indicate the reaction catalyzer. And the name of enzyme should end with the suffix "ase". "Ase" designates the enzyme, right? Now, International Union of Biology and Molecular Biology, they have classified enzymes into following 6 types. Enzymes have been classified into 6 types by IUBMB, International Union of Biochemistry and Molecular Biology. They have classified enzymes into 6 types. What are those 6 types of the enzymes? The first category of enzymes is known as oxidoreductases. Oxidoreductases. Oxidoreductases are those enzymes which catalyze simultaneous oxidation and reduction of their substrate. Oxidoreductases are those enzymes which simultaneously catalyze oxidation and reduction of their substrates. So they catalyze oxidation and reduction of their substrates. Simultaneously. For example, glyceraldehyde, phosphate dehydrogenase. This is the name of enzyme. Glyceraldehyde phosphate dehydrogenase. What is its action? It catalyzes simultaneously glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate. So we are discussing about oxidoreductases. They are the enzymes which catalyze simultaneous oxidation and reduction of their substrates. So I hope you know what is oxidation. Adding of oxygen to some substance is known as oxidation. Removal of hydrogen from some substance is known as oxidation. Removal of electrons from some substance is known as oxidation, right? So they catalyze oxidation. And you know what is reduction? Addition of hydrogen to a substance is known as reduction, right? So they simultaneously catalyze oxidation and reduction of their substrate. So we have got the enzyme glyceraldehyde phosphate dehydrogenase. What is its action? It oxidizes glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate, and simultaneously it reduces NAD to NADH. So it has got two substrates. Glyceraldehyde 3-phosphate, it is converted into 1,3 bisphosphoglycerate, and at the same time it reduces a coenzyme NAD to NADH, right? So this enzyme simultaneously catalyzes oxidation and reduction. So glyceraldehyde 3-phosphate is oxidized to 1,3 bisphosphoglycerate, while as the coenzyme NAD it is reduced to NADH. So this is the reduction reaction, this is the oxidation reaction, right? So in this way, this enzyme, glyceraldehyde phosphate dehydrogenase, it carries out simultaneous oxidation and reduction. It oxidizes glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and simultaneously it reduces NAD to NADH, right? So in this way, this enzyme, which catalyzes simultaneous oxidation and reduction of their substrates, it is known as oxidoreductase, right? This is one example of oxidoreductase. Then there is another example of oxidoreductases. Nitrate reductase. Nitrate reductase. This is another oxidoreductase enzyme. What is its action? NO, NO3 plus NADH. Nitrate reductase converts this into NO2 plus NAD plus H2O. So here, nitrate, the substance nitrate, it is oxidized to nitrite. So nitrate is oxidized to nitrite. Sorry, it is reduced. Sorry, it is reduced to nitrite. Nitrate is reduced to nitrite, right? And NADH2, it is oxidized to. This is oxidized to NAD. So this is oxidation part. So in this way, the same enzyme, nitrate reductase, it reduces nitrate to nitrites, and simultaneously it oxidizes NADH2 to NAD and water, right? So the same enzyme is carrying out oxidation and the reduction. So that is why we call such enzyme as oxidoreductases. So we have got two examples of oxidoreductases. So oxidoreductases are those enzymes which simultaneously catalyze oxidation and reduction of their substrates. So in the first example, glyceraldehyde phosphate dehydrogenase. It oxidizes glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate, and simultaneously it reduces NAD to NADH2, right? So this part is reduction reaction, this part is oxidation reaction, right? So we call such enzymes as oxidoreductase. So glyceraldehyde phosphate dehydrogenase, it is an oxidoreductase. Similarly, we have got nitrate reductase. Nitrate reductase is an enzyme which converts nitrate into nitrite. So here it reduces nitrate into nitrite. So this is the reduction reaction. And simultaneously, it oxidizes NADH2 to NAD, right? So it is the oxidation reaction. So those enzymes which simultaneously catalyze oxidation and reduction reaction of their substrates, we call them as oxidoreductases. This is the first class of enzymes. So this is the first classification of the first class of the enzyme according to IUPAC. Now we will discuss another class of enzymes. The second class of enzymes is known as transferases. We have got now another class of enzymes. These enzymes are known as transferases. Transferases. So what are these transferases? They are the enzymes which catalyze transfer of groups. Transfer of groups from one substrate to another substrate. Another substrate. For example, we have got an enzyme that is known as glutamate pyruvate transaminase. Glutamate pyruvate transaminase. So what is the function of this glutamate pyruvate transaminase? It catalyzes the transfer of groups, amino groups, from the substrate glutamate to substrate pyruvate, and it forms alpha keto glutarate, alpha keto glutarate plus alanine, plus alanine. So this glutamate pyruvate transaminase, it catalyzes the transfer of amino group. It catalyzes the transfer of amino group from substrate glutamate to pyruvate, right? And this substrate glutamate, it is converted to, it is changed to alpha keto glutarate. And this pyruvate, it is converted to another substrate, alanine, another product alanine, right? So transferases are those enzymes which catalyze transfer of groups from one substrate to another substrate. For example, we have got glutamate pyruvate transaminase. This glutamate pyruvate transaminase, it catalyzes transfer of amino group from glutamate to substrate pyruvate. And this glutamate itself is converted into alpha ketoglutarate, and this pyruvate is converted into alanine. So amino group is transferred. Amino groups are transferred from glutamate to pyruvate. Pyruvate is converted into alanine, and glutamate is converted into alpha ketoglutarate. So this is our this transferase enzyme. Hexokinase. Hexokinase. It is again a transferase enzyme. So it is involved in transfer of glucose plus ATP. It is involved in transfer of inorganic phosphate. Inorganic phosphate from ATP to glucose. So we say transferases are those enzymes which catalyze transfer of glucose from one substrate to another substrate. In this example, hexokinase transfers inorganic phosphate from ATP to glucose to form glucose 6 phosphate. To form glucose 6 phosphate plus ADP. Plus ADP. So our second class of enzymes known as transferases. They are those enzymes which catalyze the transfer of groups from one substrate to another substrate. So for that reason, we call them as transferases. The first example I cited was glutamate pyruvate transaminase. Glutamate pyruvate transaminase catalyze the transfer of amino group from pyruvate to glutamate. Similarly, another example is hexokinase. It is involved in transfer of inorganic phosphate from ATP to glucose, so that glucose 6 phosphate is formed and ATP is converted into ADP, right? So these are the two examples of transferases. So those enzymes which are involved in catalysis, which are involved in transfer of groups from one substrate to another substrate, we call them as transferases. So that is the second class of our enzymes. Then there is third class of enzymes. Another class of enzymes that is known as hydrolysis. Third class, hydrolysis. Hydrolysis are those enzymes or enzymes which catalyze the breakdown of molecules by introduction of water. By introduction of water. Hydro means water, lysis means breakdown. Breakdown. So they break compounds or molecules by the introduction of water in them. So they break the bonds in a molecule by the introduction of water in them. That is why we call them as hydrolysis. Hydro means water, lysis means breakdown. So they break the molecules or compounds by the introduction of water in them. So those enzymes which break complex molecules to simple molecules by introduction of water in them, they are known as hydrolysis. For example, all digestive enzymes, all digestive enzymes of our body. So all the enzymes which are released in our alimentary canal, they are hydrolysis. They break down complex molecules into simple molecules by the introduction of water. If you remember, amino acids they combine and they form polypeptide chains. Then polypeptide chains get three-dimensional structure and they form the protein, right? So if we have to break a protein into amino acids, how can we break a protein into amino acids? How can we digest a protein into amino acids? So at the time of formation of these peptides, polypeptides, water was removed from the individual monomers. I hope you remember how one amino acid combined with another amino acid. So one amino acid combined with another amino acid. This was one amino acid. This was our one amino acid. This was another amino acid. Then there was another amino acid. So one amino acid combined with another amino acid by the elimination of water. So this water molecule was eliminated here. And the two amino acids had joined together by the formation of peptide bond. Now, if we want to break this peptide chain, so it was formed by dehydration synthesis. This removal of water to join the molecules together. It is known as dehydration synthesis. Peptides were formed by dehydration synthesis. The water molecules were eliminated at the time of formation of the peptides. So anything which is formed by elimination of water, that is known as dehydration synthesis. Dehydration means removal of water. So when water molecules are removed to join the monomers together, that is known as dehydration synthesis. So peptides, polysaccharides, lipids, they were all formed by dehydration synthesis because water molecules were eliminated from them at the time of formation, right? Now, if we want to break this dipeptide into individual amino acids, we have to reintroduce this water molecule, which was removed from them at the time of their formation. So we cannot simply add water to polypeptide chains. If we put a protein into water, water cannot break it into amino acids. Why? Because water cannot be itself incorporated into the peptide bonds, right? So for this incorporation of water, to break the peptide bond, we need some enzymes. So those enzymes which are involved in breakdown of complex molecules into simple molecules by the help of water, we call those enzymes as hydrolysis. So in our alimentary canal, we digest proteins. Proteins are finally converted into amino acids. So how proteins are digested? They are digested by protein digesting enzymes, proteases. So how do proteases work? Those proteases, they reintroduce water into peptide bonds. So when water molecule is reintroduced at the peptide bond breaks and the individual amino acids separate, right? So this type of digestion, it is known as hydrolysis, digestion by the introduction of water, right? So all our digestive enzymes, they are hydrolytic in action. They break complex molecules into simple molecules by the reintroduction of water, right? So proteases, amylases, lipases, nucleases, they are all hydrolysis because they all break complex molecules into simple molecules by the introduction of water. So they introduce water molecules into peptide bonds, into glycosidic bonds, into ester bonds, right? To break those bonds and convert complex molecules into simple molecules. So this class of enzymes, which breaks complex molecules into simple molecules, or which breaks large molecules into smaller molecules by introduction of water, we call them as hydrolysis. The examples of hydrolysis are all digestive enzymes of our body. Amylases, proteases, lipases, and nucleases, right? They are all examples of hydrolysis. So that was the third class of our enzymes. Then there is another class of enzymes that is known as lyases. Fourth class. Lyases. Lyases are the enzymes which catalyze. They catalyze breakdown of breakdown of specific covalent bonds. Specific covalent bonds. Carbon carbon, carbon hydrogen bond, carbon oxygen bond, carbon nitrogen bond, etc. Without introduction of water. Without introduction of water. Hydrolysis also broke covalent bonds. Hydrolysis, they also broke covalent bonds by introduction of water. But lyases, they also break covalent bonds. Lyases also break covalent bonds. They break carbon-carbon bond, carbon-hydrogen bond, carbon-oxygen bond, carbon-nitrogen bond, or carbon halide bond, or carbon-sulfur bond, but without introduction of water. So what is the difference between lyases and hydrolysis? Hydrolysis also break these bonds. Hydrolysis also break carbon-carbon, carbon-hydrogen, carbon-oxygen, carbon-nitrogen bond. But they break these bonds by the introduction of water. But lyases, they do not break these bonds by the introduction of water. They break these bonds without the introduction of water. For example, we have got an enzyme histidine decarboxylase. Histidine decarboxylase. This histidine decarboxylase, it breaks histamine into, sorry, it breaks histidine. It breaks histidine. Histidine is an amino acid. It breaks it into histamine. Histamine plus carbon dioxide without introduction of water, right? So bond is broken without introduction of water. So we call this enzyme as lyase. So lyases, they are the enzymes which break covalent bonds without introduction of water. So they may also break large molecule into smaller molecule without the introduction of water. For example, histidine decarboxylase. It breaks histidine into histamine and carbon dioxide, but without hydrolysis, without introduction of water, right? Similarly, we have got another enzyme that is known as aldolase. Aldolase. This aldolase converts fructose 1,6 bisphosphate. bisphosphate into dihydroxyacetone phosphate, DHAP, dihydroxyacetone phosphate, and 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde. So aldolase, it is a lyase which breaks fructose 1,6 bisphosphate into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. Right? So it breaks the covalent bonds without the introduction of water. So DHAP, it stands for DHAP, dihydroxyacetone phosphate, and 3-phosphoglyceraldehyde, CPGL, glyceraldehyde, 3-phosphate. We call it as 3-phosphoglyceraldehyde, right? So lyases are those enzymes which convert, which catalyze breakdown of carbon, 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 hydrogen, carbon, oxygen, carbon, nitrogen, carbon, carbon, sulfur, carbon, halide bond. Or we simply say they catalyze breakdown of covalent bonds to convert a large molecule into a simple molecule. For example, aldolase. It converts fructose 1,6 bisphosphate into two compounds, dihydroxyacetone phosphate and 3-phosphoglyceraldehyde. They are both trioses, while as fructose 1,6 bisphosphate is a hexose. So it is a 6-carbon containing sugar, right? It is a hexose. This is a 3-carbon containing compound. This is a 3-carbon containing compound, right? So in this way, a hexose sugar is converted into a triose sugar, right? So they catalyze the breakdown of covalent bonds without the introduction of water. So we call these enzymes as lyases. So example of lyases is histidine decarboxylase and aldolase. These are the examples of lyases. So lyases differ from hydrolases. Both of them catalyze breakdown of covalent bonds. But hydrolysis catalyze breakdown of covalent bonds by introduction of water, and lyasis catalyze breakdown of covalent bonds without the introduction of water, right? So that was about lyasis. Then we have another class of enzymes. Then we have another class of enzymes. They are known as isomerase. Fifth category, fifth class. Isomerase. What are these isomerases? They are the enzymes which catalyze. They catalyze the rearrangement, of atoms of a molecule to form an isomer. So they are involved in catalysis of rearrangement of atoms to form isomers. For example, phospho phosphohexose isomerase. Phosphohexose isomerase. It is an enzyme. It converts, it catalyzes the conversion of glucose 6 phosphate in the presence of phosphohexose isomerase is converted into fructose 6 phosphate. fructose 6-phosphate. So glucose 6-phosphate, it is an aldose sugar. When the atoms are rearranged, then it is converted into fructose 6-phosphate, a ketose sugar, right? So this rearrangement of atoms in glucose 6-phosphate to form fructose 6-phosphate by the help of phosphohexose isomerase, right? So this is known as isomerization. And this isomerization is brought about by phosphohexose isomerase. So isomerases are those enzymes which catalyze the rearrangement of atoms to form molecules, to form isomers. For example, glucose 6-phosphate, a 6-carbon sugar, is converted into fructose 6-phosphate, a 6-carbon sugar, right? But glucose 6-phosphate is an aldose sugar, fructose 6-phosphate is a ketose sugar. So how this aldose is converted into a ketose? It is converted into a ketose by the rearrangement of atoms. So the atoms are rearranged and aldose group is converted into a ketose group by the help of this phosphohexose isomerase. So we call this process as isomerization, and it is catalyzed by isomerase enzyme. Similarly, we have got another enzyme that is known as triose phosphate isomerase. Triose phosphate isomerase. It converts dihydroxy acetone phosphate into 3-phosphoglyceraldehyde, or it converts 3-phosphoglyceraldehyde into dihydroxyacetone phosphate. So they are interconvertible. Right? So dihydroxyacetone phosphate, a 3-carbon containing ketose sugar, may be converted into 3-phosphoglyceraldehyde, a 3-carbon containing aldose sugar. Then this 3-carbon containing aldose sugar, it may be converted back into dihydroxyacetone phosphate. So these two compounds, they are interconvertible. So they are changed. Here ketose is converted into aldose, and aldose may be converted into ketose. So how they may be converted? How they are interconvertible? By the rearrangement of atoms. So what catalyzes the rearrangement of atoms? It is triose phosphate isomerase enzyme, right? Triose phosphate isomerase, which converts interconversion of dihydroxyacetone phosphate into 3-phosphoglyceraldehyde, or 3-phosphoglyceraldehyde into dihydroxyacetone phosphate by the rearrangement of atoms, right? So we call these enzymes phosphohexo isomerase and triose phosphate isomerase as isomerases, because they bring about the isomerization, right? So that is the 5th class of our enzymes. Then we have to discuss the 6th class, that is the ligases. We are running out of time, so that will not be discussed today. Inshallah, we will discuss that in next lecture. Thank you for being with me.